Hello, my name is Yordi Tamayo Molina. Today, I will introduce my recently developed app, Viral App, an application for analyzing multi-transcriptome data in the context of primary human cells infected with various viruses. Viral App was developed through a collaboration between members of the University of Antioquia and the University of Groningen. To give some background, viral infections present a major public health burden, further emphasized by the rising risk of arbovirus infections. While a wealth of omics data on viral infections has been generated, centralized databases specifically focusing on host responses to viral pathogens are just now emerging. In response, we developed Viral App using our Shiny package. The backend of Viral App currently integrates over 40 transcriptome profiles of human primary cells infected with various viruses. This data is directly downloaded from the Gene Expression Omnibus and processed within the app. The backend data feeds into the frontend, which features an intuitive interface. Viral App incorporates biological pathways to generate different analysis graphics, such as differentially expressed genes, dendrograms, heat maps, box plots, and bipartite networks. In Viral App, users can select data for analysis based on study type, cell type, virus genome, virus type, and specific virus. Users can also adjust the fold change for differentially expressed genes and choose specific signaling pathways for analysis. Currently, Viral App includes 44.7% of transcriptomes from patient samples and 55.3% from in vitro experiments involving cells such as monocytes, macrophages, PBMCs, and others like B cells and dendritic cells. These cells are predominantly infected with positive sense RNA viruses, including coronaviruses and arboviruses. Specifically, viruses like dengue, Zika, chikungunya, and SARS coronavirus 2 are covered. For example, if we apply specific filters in Viral App, such as in vitro studies with macrophages and arboviruses, or patient samples with arboviruses and dengue virus, we can organize our analyses as demonstrated here. From macrophages infected with arboviruses, we can examine differentially expressed genes and observe clustering in a dendrogram. Importantly, we see that the transcriptional signature of infected cells differs from that of bystander cells. Furthermore, bulk RNA-seq data from mixed populations of infected and bystander cells predominantly reflects a bystander cell transcriptional signature. We can explore multiple signaling pathways, such as interferon signaling, pattern recognition receptors, or stress signaling pathways. From the second filter, focusing on dengue-infected patients, we can observe differentially expressed genes in the dendrogram, which shows cell clustering according to cell type. For example, a box plot shows that the antiviral response in monocytes and PBMCs is induced, while in B cells and PBMCs from severe dengue patients, this pathway has lower induction. Conversely, the stress signaling pathway is primarily induced in B cells and PBMCs from severe dengue cases. Finally, bipartite networks show the number of genes involved in these pathways. In conclusion, Viral App was designed to explore the transcriptional programs of virus-infected cells and to generate various reports. For example, using Viral App, we can see that bystander cells exhibit a stronger antiviral response, while infected cells show a more stressed profile. Additionally, in patients, monocytes and PBMCs demonstrate genes involved in the antiviral response, while B cells have fewer of these genes. Furthermore, in severe dengue cases, genes involved in stress responses are upregulated, while those involved in antiviral responses are downregulated. Finally, Viral App is part of a larger project called ImmuneApp, which aims to develop an additional app for macrophage polarization. This work is part of my doctoral thesis and is created for the scientific community. I would like to thank Minsciences, the immunovirology group at the University of Antioquia, and the experimental virology group at the University of Groningen for their support in developing Viral App.